Hello and welcome to GeneLab Chats. Today we will be interviewing Dr. Vidya Manian from the University of Puerto Rico. A major health problem in spaceflight is muscle wastage due to microgravity. Thus, finding appropriate countermeasures to treat muscle atrophy is of the utmost importance. Manufacturing and testing of new or even repurposed drugs can be costly and time-consuming. In her publication, Dr. Manian reanalyzed 6 transcriptomic GeneLab datasets and detailed the use of artificial intelligence in identifying drug candidates as possible treatments for muscle atrophy in a rapid and cost-effective manner. This work was funded by a NASA EPSCoR call for the reuse of GeneLab data. In this interview, Dr. Vidya Manian will discuss this research and highlight how the GeneLab database helped enable this work. Welcome, Dr. Manian.
Hi.
Let me start off by asking you, what were the most important findings in this study?
In this research, we applied artificial intelligence methods combined with network science to achieve the goal of identifying drugs for repurposing for organ muscle atrophy. We combined 6 GLDS datasets to construct a huge network of genes and diseases related to muscle atrophy in organ muscle tissues such as the liver, thymus, and spleen. We call these new networks knowledge graphs. We applied the random forest machine learning method, graph neural network deep learning architecture to identify key genes of muscle atrophy in organs associated with cancer, diabetes, and neural disorders. Using link prediction on graph neural network, we identified monoclonal antibodies drug therapy for repurposing.
That's wonderful. Thank you so much for that summary. And one last question here is, how did the GeneLab database and tools help assist you in your work so you can achieve your goals?
The GeneLab database is an excellent source of curated and accurate data sets from Spaceflight. The administrators were very helpful in providing the correct data sets in a timely manner. If there are any errors, they correct it immediately and provided us with the correct data sets for gene expressions. Also, our program monitor, Dr. Egley, guided us in his research and suggested which data sets are to be used for this purpose.
Well, thank you so much for your time, Dr. Manian. This was incredibly informative. For those of you watching, learn more about this work, how to join the analysis working groups, and access all the data sets analyzed in this work with the links provided in the description section below. Thank you.